Okay, very good evening, my dear friends. This is Dr. Padala Tirupati, Assistant Professor, Department of Botany, Government Degree College for Women, Jadithya. We are started this CPGET free online coaching last 3 years onwards. So many students are get benefited. Last year also our Bhagirath sir, eminent resource person, Assistant Professor, Department of Botany, GDC Rangasaipet, Warangal district. Sir has clearly explained about the important area of the life sciences, especially biological sciences, that is genetics. It is one of the most important area for all competitive exams, including CPGET 2024 and also CUSET, JL, DL, Gurukul, PGT, and other competitive exams also. In this regard, we would like to invite Dr. G.BHAGIRATH Garu, Assistant Professor, Head Department of Botany, GDC Rangasaipet. Please welcome, sir, and start your session. Share your screen. Please welcome. Am I audible?
Hello, am I audible?
Your voice is very low, sir.
Hello, am I audible now?
Yeah, you are audible, but voice is very low. Very low voice.
Low voice, 100%. Okay, sir, now it is clear, I think.
Sir.
Hello, sir.
No. Your voice is little bit okay, but it is very low voice. If possible, increase your volume.
Okay, thank you, Padala Tirupati sir, for giving this opportunity to deliver few topics related to the botany curriculum with regard to the CPGET entrance 2024. Shall I now share my screen? Is it visible, sir?
Yeah.
Slides are visible?
Okay. Please click on slides. Okay.
It is clearly visible, I think.
Okay.
Okay, sir?
Okay. Please proceed.
Okay. Good evening, everyone. The CPGET aspirants. Today, we will discuss about one of the important topic in the botany, that is plant genetics, with related to the Mendelian principles. Today, we will discuss about Mendelian principles. Before that, every biology student should know aware about to understand the genetic concepts. First of all, every botany student know about the fundamental concepts in cell and molecular biology, that is structure of DNA, steps in replication, biosynthesis of proteins, RNA synthesis, structure of chromosome, and types of cell division occur in the living organisms. What is the difference between mitosis and meiosis, hybridization techniques and process, aneuploids and polyploids. So first of all, in this area, you know something basic fundamentals, we can clearly understood the genetics chapters. So in this lecture, we cover what is meant by genetics, heredity, and how variations comes in the life cycle of any plant, allele development, and Mendelian principles, and types of crossing, that is monohybrid and dihybrid, and other linkage or incomplete dominance related topics. So before going to that, first of all, you know the few terminology in the genetics is very important. The term indicates some meaning or some something says about the genetic experiments. So for example, every living organism shown some traits or features. So what is meant by trait? It is a characteristic that can be passed from parent to offsprings. Some characters are transmitted from parent to offsprings or next generation. So we can express in traits. So different characters. So what are the characteristics of the parents which are transmitted to offsprings through characters only? That is called traits. That whole process is nothing but is called heredity. We can call as inheritance also. The heredity also called as inheritance. So particularly in the genetics, initially from the Mendelian genetics, the main focus of the genetics in that area in those days, the study of heredity only. Heredity means what? How the parental characters are transmitted to next generation or offsprings. So only focus on that particular aspect in the Mendelian era. But according to the modern genetics, the genetics is a science of branch of biology which deals with what is the structure of gene, what is the function of gene, how the gene synthesis takes place, and how genes are modified and it changed into mutations or variations. All these things are discussed in the nowadays in the genetics branch. So what is meant by genotype and phenotype? So genotype is nothing but what is the genetical constitution of a particular trait or particular phenotype. So organisms complete genotype which is represented to a particular character. So what is the genetical constitution or genetic makeup of an organism is called as genotype. Phenotype means whether it is a physical appearance of an organism with respect to a trait. For example, we can go for the Mendelian genetics. We can always study about height of the plant, whether it is tall or dwarf or short. It is indicate that phenotyping. Phenotype means the outward expression of the particular trait. So that is called as phenotype. Color of the flower, height of the stem, or we can say example, the position of the flower, or the seed color, seed shape. All these are we can include as phenotypes. Each phenotype represented by a particular genetic makeup. That is nothing but genotype. So what is meant by dominant trait and recessive trait? A trait is expressed preferentially over another trait. It is represented in the capital letter. So when doing the hybridization process, the F1 generation plant possessing a character that is nothing but is called dominant trait. Always we can express that particular trait in the form of capital letter, that is T. For example, capital T is indicates tall character. Small t character is a recessive character. So one of the character we can't see in the FN generation of the parental character. One of the parental character is missing in the FN generation, that considered as recessive trait, that is opposite to the dominant, that is preferentially masked by the dominant gene. So it is represented by same letter but with his lower case letter and a small letter toti manam define chestam. So that is recessive trait. So test cross is nothing but it is a cross of an individual organism of dominant phenotype, but the organism with a homozygous recessive genotype. So the F1 plant is crossed with a unknown genotype, whether it is a recessive or dominant. We can test based on the progeny. We can test that the one of the parental is whether it is a recessive or dominant. That is revealed by a cross. That is nothing but is called test cross. And the reciprocal cross. Reciprocal cross means the equal contribution of the male and female gametes. We are reciprocally changed, vice versa. Any progeny ratio is changed or not. We can examine by reciprocal cross. Okay. Next one is. So genetics, we already explained about genetic studies is nothing but studying about the genes and their inheritance. For example, here these two photographs are shown here. In the rice genotypes are there. So one of the rice variety A, which is the husk color is no color. ఎండినటువంటి గడ్డి రంగులో ఉన్నటువంటి గింజలు కనబడుతున్నాయి, ఫ్రూట్స్ కనబడుతున్నాయి. In the variety B, it is blackish color. So both the varieties are belongs to rice, but the genetic makeup is different internally. That's why the variety A and variety B differ from in the color of the husk color is different. So in the same way, another genotype A and genotype B photographs are there. So which is belongs to lentil plant. So in the first photograph, we can see the seed size is bigger. In the second photograph, or genotype B is shows smaller seed size. The seed size is contributed by a particular genetic makeup internally, which is present in the DNA molecule. So how these changes are occurs, how these different parameters or different characters are contributed by the genes, and they are transmitted to the offsprings from parents. So all these are things discussed in the genetics only. It investigates genetic variations and mutations, and it's crucial for understanding the heredity, genetic disorders, and biotechnology applications also. So heredity, we already explained that the term is also called as inheritance. How these characters are transmitted. So generally, the parental characters are transmitted to the offspring either through reproductive phenomena. One is sexual reproduction, another one is sexual reproduction. In the sexual reproduction, the traits or characters are transmitted from one generation to another generation through the gametes. In the same way, asexual reproduction is also passing the characters from parents to next generation. But the mode of transmission is different in the sexual reproduction, and in the second one is asexual reproduction. For example, we can go for the how human being characters are transmitted to one generation to another generation. Every human being containing diploid chromosome number is 46, and female is also 46 chromosomes are there, 23 pairs. We can, in another way, we can call as 23 pairs. During the sexual reproduction, the male gamete or sperm cell containing haploid chromosome number is 23, and the same way female cell containing 23 chromosomes. So during the gametogenesis, meiosis takes place. Half of the chromosomes are present in the gametes. Both the gametes are carry the representative genes, and during the fertilization process, both the chromosomes are combined with each other. We are getting in the offspring or in the next generation, same number of chromosomes are appear. What are the genes are contributed by the both parents, which are seen in the next generation. So in this way, the characters are transmitted through genes via gametes into the next generation. This is the transmission of the characters through sexual reproduction. Our Mendelian principles or Mendelian genetics is applicable to those plants perform the sexual reproduction. It is applicable only. Okay. So next we can go for Gregor Johann Mendel is the full name of the Mendel person. He was born in Austria, and now it is called as Czechoslovakia in 1822. He worked
Sir,
as a
please
priest.
click on slideshow if possible.
If these photographs are
Okay,
oh,
sir.
right.
No, it is the old
Okay.
slide
Okay.
has come. Okay, sir. Okay. Yeah, it's fine.
Next. Animated, that's why it is late. Okay. So the Mendel was worked as a priest in the church. He observed the nature and plants. He developed a keen of interest to study the genetics. One of the experimental tool plant is he selected pea plant. The pea plant scientific name is called Pisum sativum. The diploid chromosome number is 14. That's why we examine 7 pairs of the characters. 7 pairs, 7 into 2, that is 14. The 14 chromosome number is diploid number of chromosomes in Pisum sativum plant. He started hybridization experiments on pea plant in 1857 by collecting pea seeds from all across the Europe and postulated Tula. He discovered 2 Mendelian principles we know very well based on his hybridization experiments. Nearly 7 long years of experimentations, he published all the results in a paper that is titled as Experiments in Plant Hybridization. So on the basis of result, he discovered the 2 main important inheritance laws. That is law of segregation based on the monohybrid experiments, and the law of independent assortment is based on the dihybrid experiments. These 2 important laws are discovered. That's why the Gregor Johann Mendel is popularized as a father of genetics. So we learn in an intermediate level also. So why he selected pea plant? So the reasons are there for that. The first one is pea varieties were easily available on commercial basis. It is a number of varieties are available in the nature. And moreover, these plants shows 2 contrasting forms of many characters. So one of the morphological character appear in 2 different plants in contrasting nature. For example, height of the stem. Some plants are grown up to several meters, above 10 meters, that are considered as tall character. Some plants are grown up to only 2 meters, that is called dwarf character. So one morphological feature shown in 2 contrasting forms. So that is very easy to carry out these experiments. He identified so many such type of contrasting forms of many characters. And the flowers of pea plant, naturally they are self-pollinated. So it is also advantage for these experiments. And moreover, this plant is an annual plant. That means few months are enough to complete its life cycle. 6 months or above the 6 months sometimes. So that's why he carried out the hybridization experiments by selecting this annual plant. And moreover, the flower size is very large and the seed size is bigger. It is also helpful for the conducting hybridization experiments and also soaking the seeds in the soil. Growing pea plants is relatively easier, and when compared to other plants, we can easily cultivate or grown very easily in the natural conditions. So, for example, the photographs are shown here. This is the important characteristic features of Fabaceae. That is the papilionaceous corolla, and the didelphous stamens are there, 9+1 stamens, which is the characteristic features of the Fabaceae members. So next we can go for what is meant by allele. So most of the students are confusion with this allelomorph, allele, genes. Allele is nothing but it is a contrast form of genes that are located at the same locus of the chromosomes. So for example, in the diploid cell, we can see 2 homologous chromosomes are there. The yellow color cylindrical shapes are there here. These are considered as chromosomes. On this chromosome, the position of the gene in a chromosome is called as locus. Here, capital P gene is there, one of the homologous chromosome containing capital P, another corresponding homologous chromosome containing small p. So these 2 genes represent 2 forms of allele. That means with the collaboration of these two genes, the particular character is manifestation or expressed in the plant. So here, trait is the physical or behavioral characteristic of an organism. We already explained. Phenotype is a set of observable physical traits. For example, in the genetical experiments, capital T, small t, we write like that. Capital T, small t. This is a allele. Both genes are called as allele or allelic genes. So, which are located on the same place in the homologous chromosome. So, same locus. That is, this is maintain the genetic information of an organism in the form of genotype. Genotype is an organism's genetic information. For example, we can see these three flower photographs are there. So first two flowers are in purple color. The purple color is determined by two genotypes, that is capital P, capital P. For example, here capital P is there. In the place of small p also, capital P is there. That considered as homozygous dominant. Homozygous dominant, it contributes which type of color? Purple. That is a phenotype. In the second case, here capital P is there on the first homologous chromosome. The second homologous chromosome containing small p. It is heterozygous dominant. Heterozygous means the two allelic genes are not similar, dissimilar. That is called as heterozygous. Two allelic genes are similar form. That is called as homozygous. సమయుగ్మజము విషమయుగ్మజము అంటాం తెలుగులో అయితే మనం. So homozygous dominant, it is contribute the purple color. In the same way, heterozygous dominant is also contribute the purple color. But what about small p, which is homozygous recessive, which it unable to synthesize some pigments. That's why the flower color is white only here. So homozygous recessive shows white color. Why? Because the representative allelic genes which may not give the information for the synthesis of a pigment. That's why it is in the white color. So genes. Gene is a basic unit of heredity and a sequence of nucleotides in DNA that encodes synthesis of a gene product, either RNA or protein. What is meant by gene? A gene is a basic unit of heredity and it is the smallest part of the DNA. DNA consisting a sequence of nucleotides in DNA that allele or gene encode for the synthesis of a gene product, either RNA or sometimes protein. So whatever the genes are present on the chromosomes, which are helps or which are contribute to synthesis of a protein or RNA molecule or a gene product. That's why any change takes place in the gene, automatically it's shown in the metabolic sequence. Alleles is nothing but two forms of a gene, whether it is dominant or in a recessive. Dominant means stronger two genes expressed in the hybrid. It is represented by a capital letter. Wherever you write capital letters, that is nothing but is called dominant allele. Wherever you write small letters, that indicates that that is a recessive gene. We can blindly remember that one. Genotype is nothing but gene combination for a trait. For example, in the Mendelian genetics, we studied capital T, capital T, capital T, small t, small t, small t, like that. So these are all different type of. So this is called allele development. Allele development what here it is, for example, we can see here. For example, a allele is there, a allele gene is there, which give the information to the RNA in the form of codon. That is codon 1 methionine and codon 2 represents glutamine. In another plant, same type of allele is there, but it is undergo mutation by the receive the UV light from the solar energy, or some toxic chemicals are present in the soil, which influence the mutation in the gene level. That's why the allele undergo mutation, which change the codon sequence also. In the first codon, ATG is here, but in the second, in the mutation, by the influence of mutation, it changed into ATC. That means G is replaced by C. So if these two amino acids are very important for the preparation or synthesis of the color. So that's why here the plant color, the flower color is purple. But here, C, the D is replaced by C. That's why both codons are not synthesize the particular pigment, unable to synthesize its particular pigment. That's why this flower is in white color. So this is nothing but allele development, how alleles are involved in the metabolic process, that is in the RNA synthesis or in the protein synthesis, like that. So in the place of methionine, we are getting here isoleucine. That's why the flower in white color. Okay. So here, think the allele here. So Mendel observed total 7 pairs of the contrasting characters in pea plant. You know very well. That is flower color, purple, white, seed shape, round and wrinkle, seed color, yellow and green, pod color is green and yellow, pod shape is inflated and constricted, and plant height, tall and short or dwarf. And another one is flower position, that is axial, and another one is terminal. So the above characters are dominant one, and the below characters are in the second column. Second row of characteristics are indicate recessive characters totally. So Mendel performed 3 type of genetic crosses, that is monohybrid and dihybrid and trihybrid. Monohybrid means, so the crossing between the two plants with selecting a single pair of contrasting characters. That means cross made between two pea plants which are differ in a single trait only. That it may be flower position or eye color or skin color. In different experiments, we can select only one trait which show two contrasting forms. That is called monohybrid cross. Dihybrid means a cross made between the two pea plants with selecting two traits. At a time, we can select two traits or examine two traits. Flower color along with plant heights, or eye color and abdomen of the color of the Drosophila melanogaster, or eye color and hair color in the human being. We can select always two traits and made a cross between the organisms that considered as dihybrid cross. And a trihybrid, the name indicates itself. Tri means 3. You can select 3 traits, or the cross made between with respect to 3 alleles or 3 different genes. So for example, we can select at a time plant size, pod color, seed shape of the sweet pea plant. It's considered as trihybrid. So in the Mendelian first law, in the monohybrid experiments, we know very well monohybrid experiments. Here, the 2 alleles of a gene present in the F1 progeny do not affect each other or do not mix up with each other. They become separate and pass into the different types of gametes in their actual form, producing 2 separate types of gametes in equal proportion. So how can we explain this inheritance law based on this see here. This is the monohybrid experiment. So first Mendel select the 2 parental plants. One parent plant possessing here what purple color, I think, for example, purple color. Here it is white color is there. So it is not a pea plant. We can consider as one example, parent 1 and parent 2. In the parent 1, flowers are in purple color. In the parent 2 plant, the flowers are in white color. So a cross made between these 2 plants, we are getting F1 generation or first filial generation under. F1 generation means after parental crossing, we are getting immediate F1 generation. So all F1 generations are in home heterozygous type forms with only one of the parental characters. Among the parents, we can observe two characters, but in the F1 generation, we select only one parental character are seen. Another parental character is masked. So based on this simple logic, a mini law is expressed by the Mendel. What is that? Law of dominance. Law of dominance. Law of dominance means what? In F1 generation, one of the parental character is expressed. Another character is missing or masked. So such type of phenomenon is called as law of dominance. So for example, you can say in the dihybrid also, we can take two characters. Among the two characters, only a couple of a pair of characters are seen in the F1 generation. Another pair is missing. So in what way we can take trihybrid or dihybrid or monohybrid? Only one of the parental character is appear, another parental character is missing. That is called as law of dominance. Next, by selfing the pollination, by selfing of F1 generation, we get F2 generation. This is F2 generation. So in the F2 generation, phenotypic ratio, we are always getting 3:1 ratio, and the genotypic ratio is 1:2:1. So see here, this is the Punnett square or checkerboard. So from the F1 generation, we are getting two different gametes. So in the F1 generation, see here, two alleles are there, two allelic genes are there. What is capital P and small p? So at the time of gamete formation, these two genes are separated. But in the F1 generation, these two genes are present, but they do not mix up with each other. They are always independent. They do not mix up with each other. They do not blend of its characters. So that's why in the F generation, we are getting two parental characters. In the F2 generation, we can see parental characters. But in F1 generation, we can see only one parental character. What about other parental character? Actually, it is present in the F1 generation, but it is not expressed. That is called due to the law of dominance. The capital P gene masked the character of small p gene. That's why the small p gene, it never lose its character. It is represent in the F2 generation. So that's why law of segregation means what? So in the F1 generation, heterozygous dominant alleles which do not mix up with each other, or it may not blending with each other, at the time of gamete formation, they separate and enter into the gametes, and they randomly united with each other, and we get a F2 generation. So this is the, based on it, we can explain about the first law of Mendel's. Okay. So this is the male gamete and female gametes. By the fission, we can see like this. We are getting the flowers. So three purple colors are there. That's why three purple and one white. That is the phenotypic ratio is 3:1 always in the monohybrid. And the genotypic ratio is 1:2:1. One is homozygous dominant. Remaining two are their corners. Diagonally, we can see that capital P, small p, capital P, small p. Two are there. So these are also in the purple. But last one is small p, that is homozygous recessive. So this is the genotypic ratio. Among the 7 characters, we can always obtain 3:1, 1:2:1 only. So we can go for the second, that is dihybrid experiment. dihybrid experiments. In the dihybrid experiments, we can select two pairs of contrasting characters with respect to the shape of the seed, that is round and wrinkled, and another one is color of the seed, that is yellow and green. So yellow and green and round and wrinkled. So both the characters are examined at a time in the parent plants and select the two homozygous parental plants. And in the F1 generation, we are getting heterozygous form. And among the four characters, we can see only two, that is round and yellow. So remaining two characters are missing. It is also follow the law of dominance. Whether it is trihybrid or dihybrid, law of dominance is always present. So by selfing the F1 generation, we are getting the F2 generation. So in the F2 generation, we get four different gametes. By union of these gametes, we are expecting the F2 generation in the 9:3:3:1 ratio. 9 plants shows round and yellow character. 3 plants are shown round and green, and 3 plants are shown yellow and wrinkled, and 1 plant shows green and wrinkled. This is the last one. So these are all yellow. Total, you can see this 9, and these are the 3 green and round. These are the 3 yellow and wrinkled. So based on that, we can explain that how individually we can express it in a different form. See here. So for example, we can go through the segregation of law of independent assortment. How can we explain by this dihybrid experiment? See here, round character and yellow character. We can go through the round character. It is monohybrid. We are getting 3/4, 1/4. Ratio is 3:1. We can say only yellow. These are the 3 yellow plants. One is the green plant. But we can choose the dihybrid round and yellow. Round and yellow. Round is here. How many are there? 3/4. And yellow is how many are there? 3/4. 3/4 into 3/4. How much it is? 3/4 into 3/4, 9/16. What is that? 9/16. And here, second the phenotypic round and green. Round is how many here? 3/4. Green is how many here? 1/4. 3/4 into 1/4. How much it is? 3/16. In the same way, wrinkled yellow. Wrinkled how many here? 1/4. Yellow is here 3/4. 1/4 into 3/4. How much it is? 3/16. And last one is wrinkled. What is that wrinkled here? 1/4 part. And green is 1/4. 1/4 into 1/4. How much it is? 1/16. So the law of independent assortment says that a pair of genes which may not linked with another pair of genes, which is easily or freely segregate and enter into the gametes and form different random union takes place. Different chromosomal arrangements, but these are the, we can represent by the chromosomes arrangements also. So this is about the dihybrid cross and a monohybrid cross. So in the monohybrid dihybrid cross, we can explain two main important laws, that is a law of segregation and law of independent assortment. Law of independent assortment explain about a pair of genes or a pair of allele which may not linked with another pair of genes which are independent from each other. A pair of genes during the gametogenesis or during the meiosis, they freely enter into the gamete, and which receive only one factor or one allelic gene into the gamete. Prati gamete lo one allelic gene maatrame untundi, rendu undavu. Ade basic fundamental point. Okay, next one is cross is the cross between the individual with the unknown genotype of the individual with homozygous recessive type. So we can test the genotype of the one of the parental plant. We can that plant crossed with homozygous recessive genotype. So it indicates that 1:1 ratio, that is test cross ratio. If it is dihybrid, we can 4:1, 1:1:1:1. Four parental types are seen in the test cross. All are equal ratio getting in the next progeny. So based on that, we can say that is the test cross is the between individual with the unknown genotype. We can know the unknown genotype whether it is the homozygous dominant or heterozygous dominant. So we can easily find out by the test cross process. So this one is very important in the examination point of view. So it is a general formula for how many gametes are formed from the particular genotype. Number of different gametes produced by the F1. So we can apply this formula 2 to the power of n. Definitely we can calculate the number of gametes are formed from the particular genotype. Number of individuals in the perfect F2. In the monohybrid, how many phenotypes we have achieved? 4. That's why 4 to the power of 1 we are getting 4. For example, in dihybrid, 4 square, 16 individuals are getting in the F2 progeny. If it is trihybrid, 4 cube, 4 4 is 16, 16 4 is 64. Total 64 genotypes, 64 individuals are obtained in the F2 generation. So F2 generation లో వచ్చేటువంటి సంతతి సంఖ్య అన్నప్పుడు మనం ఈ ఫార్ములాను వాడతాము 4 to the power of n. సో అది మోనోహైబ్రిడ్ అయితే ఎంత, డైహైబ్రిడ్ అయితే ఎంత, ట్రైహైబ్రిడ్ అయితే ఎంత అనేది మనం చెప్తాం. నంబర్ ఆఫ్ డిఫరెంట్ జీనోటైప్స్ ఇన్ ద F2 జనరేషన్. ఫర్ ఎగ్జాంపుల్ F2 జనరేషన్స్ లో మనకు ఎన్ని రకాల జీనోటైప్స్ వస్తాయి? ఫర్ ఎగ్జాంపుల్ ఇన్ ద మోనోహైబ్రిడ్ we are getting 3 genotypes. One is homozygous dominant, another one is heterozygous dominant, another one is homozygous recessive. 3 kinds of genotypes are seen in the F2 generation. So by this formula, we can number of different genotypes are seen in the F2 generation by apply this formula 3 to the power of n. Number of different homozygous genotypes in the F2 generation 2 to the power of n. Number of different homozygous genotypes. For example, in the monohybrid, how many homozygous different plants are formed? 2 only. One is what is that? Tall. Another one is dwarf. Tall is capital T, dwarf is small t. So 2 to the power of 1, we can write 2 only. In case of 2 square, so number of different homozygous genotypes are in F2 generation, and we can apply this formula. So in all formulas, we can see n. N is refers to number of genes segregating. Number of alleles are segregating. Based on that, we can identify all these numerical problems are solved by these formulas. So next part is we can go for the linkage part, and in the next class we can discuss about that one. And dominance is our extension of Mendelian principle explained by 3 types of dominance: complete dominance, incomplete dominance, codominance. So incomplete dominance is nothing but here the partial expression of the gene in the F1 generation, it completely differs from the parental characters. So based on that, we can explain that in 1:2:1 ratio we are getting in the F2 generation, either phenotypically or genotypically. So incomplete dominance means here parents are 2 colors, red and white, but in the F1 generation we are getting purple color. So why it is called purple color? The partial expression of the dominant gene in the heterozygous forms, it shows a different character, that is intermediate character. Intermediate character means white and red mixed, we are getting purple color. So the partial expression of the both the genes in the F1 generation, we are getting an intermediate character or intermediate feature of the parental plants. So F1 generation, we are getting F2 by selfing. It is 1:2:1 ratio genotypically and phenotypically. By doing this, conducting the experiment called Corens in the Mirabilis jalapa, that is 4 o'clock plant. So, okay. So this is about today's class. Law of dominance, law of segregation, law of independent assortment. Is it clear? Hello? 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 Hello?
Okay,
Hello.
Bhagirath sir,
Uh,
shall we wind up the session?
just 2 minutes. By today's class, we have focused on some important questions. Generally, in the PG entrance examination, asked about the ratios. What is the phenotype and genotypic ratios of monohybrid and dihybrid, along with the test cross also. But why Mendel is success in his experiments? Why? Because he selected only 7 pairs of the characters, the responsible genes which are arranged on different chromosomes. That's why it is not linked genes. These genes are not linked. So that's why Mendel is successfully carried out his experiments with similar type of ratios. He select any character. అతడు ఎంచుకున్నటువంటి 7 జతల లక్షణాల్లో ఎప్పుడు ఏ లక్షణాన్ని తీసుకున్నా కూడా అతనికి సిమిలర్ గా రేషియో రావడానికి రీజన్ ఏంటి అంటే 7 జతల లక్షణాలకు సంబంధించిన జన్యువులు వేర్వేరు క్రోమోజోమ్ల మీద ఉండడమే అని చెప్పొచ్చు. ఒకటి, ఇవి లింక్డ్ జీన్స్ కాదని మనం చెప్పొచ్చు. సో ఎందుకు ఇతడు మెండల్ పీ ప్లాన్ సెలెక్ట్ చేశాడు అనే క్వశ్చన్స్ అడుగుతారు. తర్వాత రేషియోస్ మీద ఎక్కువ క్వశ్చన్స్ కవర్ చేయడానికి అవకాశం ఉంటుంది. మెండల్ ఫాదర్ ఆఫ్ జెనెటిక్స్ అని ఎందుకన్నాడు, లాస్ మీద క్వశ్చన్స్ ఎక్కువ అడగడానికి స్కోప్ ఉంటుంది. ఫర్ దిస్ కంక్లూజన్ we can close the topic, today's topic. Okay, sir.
Okay, thank you very much, sir. Really, it's a extraordinary and exemplary class because of plant genetics. It is one of the most important area, and also sir has clearly explained about the basic information along with Mendelian principles and monohybrid cross and hybrid cross, along with some of the important terminology regarding of this genetics, especially alleles, okay, and genotype, phenotype. These are the most important terminology, and they are homozygous or heterozygous. Maybe the alleles are homozygous condition or heterozygous condition, or even though the recessive and dominant. These are the most important terminology in the plant genetics and also the life sciences. So really, it's a wonderful class. So tomorrow, same time, we will going to meet in this class. Okay. Thank you.
Thank you.